Hello, my name is Patrick Blevins, and I will be discussing the regulation of the complement system. This is for exam 2D. I'm going to start off just with a quick overview. The complement refers to a set of serum proteins that cooperates with both the innate and the adaptive immune systems to eliminate blood and tissue pathogens. Research of the complement system began in the 1890s when Jules Bordet showed that sheep antiserum to the bacterium Vibrio cholerae cause lysis or membrane destruction of the bacteria, and that heating the antiserum destroyed its bacteriolytic activity. The complement system is an important component of the innate immune system that is crucial for the defense from microbial infections and for clearance of immune complexes and injured cells. In normal conditions, complement is tightly controlled by a number of fluid phase and cell surface proteins to avoid injury to autologous tissues. There's more than 30 components and regulators, or more than 30 components and regulators have been identified that are widely distributed in the circulation and in tissues where they are synthesized and secreted by a number of cells under various stimuli, including cytokines and hormones. Despite the lack of specificity that characteristics the components of the acquired immune system. Complement recognizes selectively foreign pathogens and damaged cells using the recognition molecules of the classic lectin and alternative pathways. The next slide we're going to talk about is complement activity is passively regulated by protein stability and cell surface composition. Protection of the vertebrate host cells against complement-mediated damage is achieved by both general, passive, and specific active regulatory mechanisms. The relative instability of many complement components is the first means by which the host protects itself against inadvertent complement activation. The powerful effector functions of complements have the potential to harm the host. The activation of CP and LP is largely dependent on foreign material, but under certain situations, like in tissue ischemia and reperfusion, both pathways can be activated and cause autologous injury. More relevant to complement-related human diseases is deposition of C3b via AAP activation, and amplification is non-discriminatory, and if not properly regulated, can damage host cells rapidly. Thus, this is essential that activity be downregulated on host cells while efficient activation is permitted on foreign targets. Human beings and other mammals have developed a variety of both plasmatic and membrane-bound inhibitory proteins to regulate the location and the activity of complement. I did list some proteins that are involved in the regulation of complement activities. Number one, I put for the C1 inhibitor. We'll go back in a minute and discuss their functions and what pathways they affect. Number two is the decaying accelerating factor, or DAF, or CD55. Three is CR1, or CD35. Number four is the C48BP. Five is factor H. Six is factor I. Seven is membrane cofactor of proteolysis, MCP, or CD46. Number eight is the S protein, or the vitronectin. Number nine is protein, or CD59. And number 10 is carboxypeptidases, N, B, and R. So looking at C1 inhibitors, the pathway that's going to be affected by that is going to be either the classical pathway or your lectin pathway. Its function is that it induces dissociation and inhibition of C1R2S2 from C1Q. It's a serine protease inhibitor. Number two, the decaying accelerating factor, or the CD55. Uh, it can affect either the classical pathway or the alternative and lective pathways. Its function is to accelerate dissociation of C4B2A and C3BBB C3 convertases. Number 3, which is your CR1 or the CD35. Uh, it also can affect the classical alternative in lectin. Its function is it blocks formation of or accelerates dissociation of the C3 convertase, C4B2A, and C3BBB by binding C4B or C3B. Number number 5 is the factor H. Its pathway that it affects is alternative, and its function is to blocks formation of or accelerates dissociation of the C3 BBB C3 convertase. The factor I's can block all 3 pathways as well: classical, alternative, and lectin. It's a serine protease. It cleaves C4B and C3B using cofactors shown. Um, the next one we're looking at is the membrane cofactor, or I'll just call it CD46. It too affects all 3 pathways: the classical, the alternative, and the lectin. Its function is as a cofactor for factor I in degradation of C3B and C4B. Number 8 is the S protein. It can block all pathways. It binds soluble C5b67 and prevents insertion into host cell membranes. Number 9 is the CD59 or protectin. It binds C5b678 on host cells, blocking binding of C9 and the formation of the MAC complex, and it affects all pathways. And the last one, the carboxypeptases, N, B, and R. The pathways it affects are the anaphylactoxins produced by all pathways, and its function is to cleave and inactivate the anaphylactoxins C3A and C5A. The last thing we're going to look at is complement system dysfunction. The complement system is thought. Sorry about that. 1 second. Sorry about that. The complement system is thought to play a role in many diseases with an immune component. It is the cause of many autoimmune diseases. When system is hyperactive or is subject to dysfunctional regulatory proteins, it drives a severe inflammatory response in numerous organs. The kidneys seem to be particularly vulnerable to complement-mediated inflammatory injuries. Injuries may derive from deposition of circulating active complement fragments in glomeruli, but complement locally produced and activated in the kidney may also have a role. Many kidney disorders have been linked to abnormal complement activation, including immune complex mediated glomerulonephritis and rare genetic kidney diseases, but also injury associated with progressive proteinuric diseases or ischemia reperfusion have also been reported. So there are some dysfunctions of the complement system, and they can uh, turn on the body. Basically, they go from being the defense for our body to the aggressors against our body. The last thing is my references. This is what I use to do this video, and I am found Dr. Blair for you using this in your library. Thank you.